Let's take a look at the carbohydrate. Carbohydrates, also called sugar, starch, glycogen, and cellulose, are polyhydroxy aldehydes and ketones, or compounds that can be oxidized to them. First, let's take a look at the monosaccharides. The simplest carbohydrates having 3 or 6 carbon atoms. A carbonyl group is either at C1 or C2 position, or the remaining carbons bound to a hydroxy group. The first way, monosaccharides with the carbonyl group at C1 position are aldehydes called aldoses. The second type, monosaccharides with a carbonyl group at C2 position are ketones called ketoses. Here we have 3 examples. The first one is called glycogen, 3 carbonyl group at C1 position. The second one is dihydroxyl acetone. Also is 3 carbon sugar. The carbonyl group at the C2 position is a ketone, is a ketose. The third one is glucose. Glucose aldose is a 6 carbon, the carbonyl group at the C1 position. Glyceraldehyde and dihydroxyl acetone, the 2 simplest 3 carbon carbohydrate are isomers. This is the formula C3H6O3, which are only carbohydrates with the name not ended in suffix OSE. All other monosaccharides are named with the OSE ending, for example, glucose. Also, we list 2 structures of ribose and ribonose. Ribose has 5 carbons, ribose. Ribonose also has 5 carbons. You can see they are isomers. The configuration different at C2 position. A monosaccharide is characterized by a number of carbons in the chain. 3 carbon sugar called triose, 4 carbons tetrose, 5 carbons pentose. The striking feature of carbohydrates is the presence of chirality centers. All the carbohydrates except dihydroxyl acetone contain 1 or more chirality centers. Let's take a look at glyceraldehyde first. Glyceraldehyde is a 3 carbon carbohydrates with only 1 chirality center. So there are 2 enantiomers. One is called enantiomers, natural occurred, and L-enantiomers. The C2 is the chirality center. It's a C1, it's a C2. This is a formula structure, 3-dimensional formula structure. This is the Fisher projection for 3D formula. This carbonyl group on the top, the methyl group at the bottom, the hydroxy group on the right, the hydrogen on the left. This is the D configuration, D enantiomer. Second, let's take a look at the monosaccharides with more than one carotid center. The Fisher projection formulas are used for compounds like aldehydes that contain several carotid centers. This is the glucose. Glucose contains 4 carotid centers: C2, C3, C4, C2, C3, C4, C5. The configuration of carotid center farthest from the carbonyl group determine whether monosaccharides are D or L. The farthest, this one, the carotid center of C5, the farthest from the carbonyl group. In Fisher projection formula of glucose, C5 is the carotid center for determining glucose configuration. Hydroxy group on carotid center of C2, 4, and 5 on the same side. The C2, C4, and 5 on the same side, and the C3 on the opposite side. A D-glucose has the C5 hydroxy group on the right. L-glucose has a C5 hydroxy group on the left. All the naturally occurring sugars, including glucose, are the D configuration. Let's take a look at the cyclic forms of monosaccharides. Formation of a stable cyclic hemiacidone and anomeric carbon. The first one, cyclic form of D-glucose. Glucose undergoes intramolecular cyclization by forming hemiacidones, while in the interaction between C5 hydroxy group and the carbonyl group, the carbonyl carbon becomes anomeric carbon. The carbonyl carbon becomes anomeric carbon after cyclization. There are 2 cyclic forms of D-glucose. One is alpha anomer, another one is beta anomer. The flat 6-carbon ring are drawn in Haworth projection. The alpha anomer has the hydroxy group on the anomeric carbon drawn down below the ring. The beta has the hydroxy group of anomeric carbon drawn up above the ring. Take a look at this. This is a rearranged cyclic D-glucose. It's not linked. It's a cyclic. Then after the cyclization, they form 2 forms. One is alpha, the hydroxy group on the anomeric carbon, the first carbon 1, draw down. This is the alpha. This is the beta. Beta is this hydroxy group draw up. Let's take a look at steps of drawing Haworth projection. The step 1. Place the O atom, this O atom, in the upper right corner of the hexagon ring, and the hydroxy methyl group, this one, on the first carbon to the left of the O atom. Okay, the first carbon here. And step 2, place the anomeric carbon, this carbon, on the first carbon as a first carbon, clockwise from the O atom. Step 3, add the substituents OH or H to the 3 remaining carbons. The 3 remaining carbons. Carbon 2, carbon 3, carbon 4, clockwise. The substituents on the right of the Fischer projection are drawn down in Haworth projection. Substituents on the left Fischer projection are drawn up in the Haworth projection. Next, let's take a look at cyclic form of fructose, a ketone hexose. Fructose forms a 5-member ring when it cyclizes to a hemiacetal because the carbonyl group at C2 instead of C1. Here, this is C1, this is C2. The cyclic form has 2 hydroxyl-methyl groups, one of which is at anomeric carbon. This is the fructose. This is the rearranged fructose. And you can see they cyclized. They form 2 forms. One is called D-fructose. Another one is called alpha D-fructose. Another one is called beta D-fructose. Alpha D-fructose, this hydroxy group on the anomeric carbon is down. This one is up. So this is the beta, this is the alpha. Next, let's take a look at the disaccharides and the polysaccharides. Disaccharides are carbohydrates composed of 2 monosaccharides. Disaccharides are acetose, compounds that contain 2 alkoxyl groups bonded to the same carbon in forming a disaccharide acetal, a hemiacetal of 1 monosaccharide interact with the hydroxy group of another monosaccharide. The new CO bond that joins the 2 rings together is called a glycosidic linkage, glycosidic bond. Take a look at this. This is 1 monosaccharide, this is the second monosaccharide. This hydroxy group on the anomeric carbon interact with another hydroxide group with on another molecule, and they form this disaccharide and water. Glycosidic bond can be oriented in 2 different ways. The alpha glycoside has a glycosidic bond oriented down below the plane of the ring. The beta glycoside has a glycosidic bond oriented up above the plane of the ring. The notation 1 to n is used when n is a number to indicate which carbon of the following monosaccharide is bonded to the acetal anomeric carbon C1 in the glycosidic bond formation. Take a look at this. Okay, this is 1 to 4. It's a 4 position. Glycosidic bond. This is alpha glycosidic bond, because this bond is below the ring, the plane of the ring. This is a beta configuration. This glycosidic bond above this sugar ring. Hydrolysis of disaccharide cleaves the glycosidic bond and forms two monosaccharides. The disaccharide can be hydrolyzed into two single monosaccharides. Okay, this bond, the glycosidic bond, can be hydrolyzed. The last one, let's take a look at the polysaccharides. Polysaccharides have several types. First one is starch. Starch is the main carbohydrates in corn, rice, and potatoes. Starch is the polymer of repeating glucose units joined by alpha glycosidic bond. You can see this bond is below the sugar ring. Also, this is the main chain. Also, you can see the branches here. Glycogen is the major form in which polysaccharides are stored in animals, like starch. Glycogen is a polymerized by glycosidic bonds by alpha glycosidic bond. Glycogen is extensively branched. That's the difference between glycogen and starch. Starch is less branched. The last one is cellulose. Cellulose is a unbranched polymer of repeating glucose unit joined in 1,4 beta glycosidic bond. This is the beta. Cellulose is cleaved by an enzyme called beta glycosidase. Humans do not possess this enzyme, therefore cannot digest cellulose. Ruminant animals like cattle contain bacteria that produce the enzyme in their digestive system, therefore can use grass as a food. This is the structure cellulose. They look like this. Okay, this is a glycosidic bond. It's in beta configuration. Lastly, let's take a look at the reactions, chemical reaction of monosaccharides, the reduction and the oxidation. The first one, reduction of aldehyde carbonyl group. This carbonyl can be oxidized. They become a hydroxy group. Carbonyl group of aldoses is reduced to 1 degree primary alcohol using hydrogen in presence of this metal is a palladium PD. So this one is reduced to the oxidation. Oxidation of aldehyde carbonyl group. The aldehyde carbonyl of aldose is easily oxidized with a variety of reagents to form a carboxy group, become the acid, yielding the adonic acid. Glucose is oxidized with this called a Benedict reagent, the cupric compound containing oxidizing agent. Cupric containing oxidizing agent. This is oxidation. The carbonyl group oxidized become a carboxylic group.